Okay, now we're starting. Okay. Thank you guys for coming out. This will be the chair that someone else can sit in. Thank you guys for all being very excited about decentralized science and New York City and meeting other science people that happen to be here. So I think a lot of people are curious, like, what is DeSci? Maybe you're, and so at the very highest level, decentralized science is about how you can use the blockchain to improve some of the various aspects of science. I'll give a little talk around that, but I just wanted to welcome everyone. And we try and do these once a month. It's a very grassroots effort here in New York. I see a lot of people taking pictures. We're trying to grow our Instagram and our Twitter a lot, so if you tag us at DeSciNYC on Instagram or Twitter, surprisingly that name wasn't taken for either of them. I would appreciate it, but no pressure. Okay. Okay, so I'm just going to give a little talk around what is decentralized science, and then we'll have our main speaker. So, what is DeSci? So, I did a PhD before, and it was sort of around traditional science and computer science. And as it was finishing with it, I was taking a lot of notice around the blockchain and how the blockchain was being used for things like for Bitcoin to exchange money and value. And then that sort of around that same time also Ethereum came into fruition, which was around doing a distributed computation, the ability to do distributed computation and trusted computation. And what those were really useful for were for decentralized finance. So you could figure out, people could exchange value with each other and build all these lending protocols on top of blockchain and Ethereum. But one of the problems with this is there was nothing that was it was all just this meta stuff. There were these colored coins where someone had a red coin and you had a blue coin, and you could exchange value, and the red coin was worth more because the team that did it was really smart. But at the end of the day, there was nothing that was sort of backing that besides just reputation in some ways. And one of the things that's really cool about decentralized science is you can use all these really powerful distributed technologies, but instead of having them backed by just red or blue coins, you can back it with actual extrinsic knowledge that is being taken from the real world and put onto the blockchain, and then people can trade those knowledge coins, or they can verify computation, or they can verify data sets, and you're using sort of all this really cool decentralized technologies to improve science as opposed to just sort of swapping tokens for decentralized finance. So the goal of all this is to improve and create a more transparent, a more open accessible, and a more collaborative scientific research environment. Okay, so the problem is the way science is done right now is so baroque and broken because it really hasn't adapted too much since the 1500s. Let me just make sure we're recording.
You're good.
We're good?
Obviously.
Okay, good. I had a lot of people who weren't able to make it, but they want to see it. Okay, so the problem is when people are doing science today, it's actually very centralized. So if you want to get money, if you want to do science, the first thing you have to do is you have to apply for a grant. And the people that you have to apply for the grant from is typically a government agency. But the problem with that is the government agency, they have if they start off as the people who are giving the grant is 30 years old, they just give the grants to the people that are always their friends. So there's not a lot of new influx of ideas. You can look at the cohort, it's the median age of the people that get the grants and give the grants is all the same, and it just kind of moves forward and forward and forward. So this is not great, because it doesn't allow for the introduction of new ideas. The other thing that sucks about it is it's slow. So publications can take 6 months or a year and a half before they're ever released in a big journal. And it's also it puts a lot of limiting factors. So they'll give you all these recommendations of things you need to do to fix it. And by the time the paper is done, there's not the core kernel of the idea anymore that's left. It's kind of sort of an amalgamation, which sucks. And I think this is actually the biggest one. This next one is like the reproducibility and replicability in science. So this is like a looming threat that is going to come, and we're seeing it now more and more. The president of Stanford had to resign because the data was sort of falsified, and they couldn't trace it back. And then the president of Harvard was plagiarizing data. And I think in general, if at the top of these institutions the people are having these problems with reproducibility, it probably trickles down. And I think the analogy would be is like, if Lance Armstrong is doping, it's very likely that a lot of the people under them have to dope as well in order to remain competitive at that same level. So I think that it's very likely that we're going to have a lot of problems with reproducibility in the future. And we're just sort of seeing the tip of the iceberg at it now. And then lastly is like complex IP agreements. So ideally, like people that do science in a university setting should be incentivized to do the science and should be able to reap some of the benefit of that. But the problem is like a lot of the IP that's created in universities gets stuck between the university and the department and the PI, the principal investigator, and the graduate students. And it's not easy to track who did what and who contributed what. And this is really bad because ideally the person that does the most of the work should gain most of the value. Okay, so how does blockchain fix this? Is now blockchain has a decentralized way for people to get money. Blockchain has democratizes more people to become involved in science, and you can track where the data comes from and who contributed what, so that everyone is rewarded properly. And it engages a lot more people through crypto economic incentives. So it's not just about like decentralizing science, but it's also about incentivizing science in the right way. I mean, one of the problems that was also with the publishing platform was like the vectors in which the money flows is kind of backwards. So to get your paper accepted, once the paper is accepted, you actually have to pay the journal. And then to get access to that paper, like they have to pay the journal also. But the journal should really be paying the authors and the people that review, and the reviewers don't get any money. So there's a lot of incentive mismatches, which lead to basically people like giving their friends things, and then like when the incentives aren't clear, like you get these like weird outgrowths of incentives where people do things that they probably shouldn't be doing with the goal of some other goal of getting a professorship or some sort of plum appointment or something else. So it's really neat to like be able to get rid of all that and just sort of start fresh and say like, okay, this is interesting. And the other thing I want to mention is like a lot of these problems could be fixed at the social level, but like there is no impetus to fix it at the social level. So we have to come up with these very complicated technologies in order to fix the social thing. So another example would be like Bitcoin. Like we could just like stop printing money, but like that's not going to happen at the social level because it's way too hard to fix it. So it's much easier to just sort of create a new blockchain where people can like trade money and create a new system. Even though it's really complicated technically, it will lead to like a better outcome. Okay, this is a slide. We already talked about it. What's cool about DeSci is there's all these new initiatives. There's DAOs that are being formed, which is a decentralized autonomous organization, which is like a lab in the traditional sense. And people are finding funding through new ways of crowdsourcing. People are publishing on-chain and doing peer review in new and interesting ways. IP is being tracked in new ways. And DAOs are sort of driving new types of scientific innovation. This is more about the funding. People are publishing new things. People are doing new types of publishing. People are publishing on IPFS. People are attaching reputation to wallet addresses so that they can see who's really good at predicting which paper is going to be successful or not. So people can create people are creating a prediction market around publishing. So instead of just someone saying like, oh, this is like a good paper, people are staking like $100 or $1,000 on a paper and saying, I bet in a year or two this paper is going to be worth more. And so they're being incentivized to do a good job of figuring out which papers are going to be successful. Another really cool thing is like tracking of intellectual property. So you can start from the beginning of where the data comes from on the sensor, and you can track all the way through up to the final published paper. And what's cool about this is now you can start to incentivize the people that are actually doing the contributing the data or actually doing the analysis, and each person sort of leaves a mark on that on each piece of data, and it allows them to be incentivized at the end. So I think there's some challenges though, definitely for decentralized science. But in traditional science, you have a very clear source of funding, but in decentralized science, it's almost like it's more broad, so you have to put a little bit more effort into finding the money. Another hard one is I think decentralized science works very well for longitudinal research, where everyone contributes a small amount. But I think it becomes much harder to do decentralized science, where you need someone with a lot of domain expertise to really understand how the Krebs cycle works, or how a very complicated thing that requires a lot of knowledge, because there's not a lot of people who are willing to put in all the energy to do that. And the university does a really good job of screening for those people who are willing to put in the energy. And then lastly, regulatory things, I think one of the things we have to look out for is people doing things that are in compliance, that are generally socially acceptable. So in a university setting, there's an IRB, which stands for an Institutional Review Board, that makes sure what the studies that are being done or conducted are generally ethical. And I think we need to be careful about that in decentralized science also. So I think we have that has the ability to revolutionize new areas of science with some caveats. And I just wanted to talk a little bit more around, so that's what decentralized science is. So DeSci, we do these monthly. And here's the next DeSci event. It's going to be on focused ultrasound. So if you scan this, you can RSVP on it. And focused ultrasound is very interesting. It's basically it's like the cyberknife, where you're trying to resonate some certain area of the brain at a very specific frequency so that the cell sort of explodes. And it's completely non-invasive, and it's looking very promising. There's some people at UCLA who are looking into it. So that is our next DeSci event, which will be in February. Yay. That is that portion of the talk. Does anyone have any questions before our main speaker?
I have a question. You said that issues the NFT when you do the data. What is the NFT? Is just simply the proof of that you attributed data to link to, or is there's some other certificate process with it?
There's an attestation from our server to that, and it also has the data on it too. So the data is stored on IPFS, and then the IPFS ID is linked to that NFT, and so then it's all there.
Gotcha.
Yeah.
For the original sale, how is that price decided?
There's no price decided initially. You're minted the NFT, and then that price fluctuates based on the value of the data set or the market. Yeah.
You see potential for all this to be integrated into university setting, because I think one of the biggest issue with research is having facilities and backup resources.
Yeah, it's a good question. I think there's going to be something like a cloud kitchen model for there's already been people that have approached DeSciNYC who have extra lab space that they're not using and are willing to rent it out to people. So if you wanted lab space, we actually have people, depending on what it is. It was like some wet lab space. So I don't think the wet, and in university settings, you can actually a lot of times rent out bench space as well. And they'll do it for you as a for-profit thing. So I don't think the physical plant of the university is a huge barrier to actual science. It's a good point, though. Yeah.
You mentioned one of the problems with the status quo of research amongst competition is there's also bias.
Yeah.
And a lot of that bias that you alluded to comes from sort of cliques, or, your friends and family, the scientific community, supporting decisions. So, any thoughts on how DeSci can avoid creating bubbles like this as well?
Yeah, I think it's a good point. I think, and definitely in Web3 in general, there are DAOs that are very cliquish, and probably even more cliquish than some of the labs in some universities. And so maybe that won't be resolved, but there'll be more. So you will be able. There's nothing per se wrong with being cliquish or unique, as long as there's a cliquish and unique place for each person who wants to participate. Like in some ways, like being unique is like if everyone is unique, that's kind of like the best outcome in the world, because everyone has their own domain in which they're not competing, and it's like a hetero-optimal situation. I think there's just like there'll be more opportunity, but it is something to be worried about, and I do think that like Web3 is generally not great at creating like groups of people and letting new people in. I think there should be like maybe like an entrance test or like something like that.
To me, the biggest downside of being cliquish is exemplified in the longevity field.
Yeah.
itself.
Yeah.
Where a lot of resources and attention is really, if you look outside that, every other biologist and scientist is going to
Yeah. Yeah. Science is very cliquish just in general. Everyone has their idea, and there's this saying that like death is the only way that new ideas get admitted into science, because the only way for a new idea, someone has to sort of physically die before the new thing is allowed in. And I think it's kind of true. And maybe just people don't communicate as much, and you'll have less conflict. But it's a good point. It's a very good point. Anyone else? Okay. All right. Well, before we do that, let's get a group picture. So everyone, try and gather in. 3. Here on the side, come in. Come in the side. We can't see the people on the side. Okay. 3, 2, 1.
Ow.
Say cheese. Okay, one more. 3, 2, 1. Cheese. Yay! DeSciNYC.
Mikey, it's on. Do you want to put this?
Oh, here, come on. You do it.
I didn't know
We have our
how live this
professional
camera
photographer.
Yeah, Allison!
Okay.
So now this is the actual one.
Okay. I'm standing on here for a second. Okay. Okay. 3, 2, 1. Okay, I think I got everybody. Perfect.
You're officially a professional photographer. Yes, you do it. Yeah. That's not charging eventually.
Michelle.
Yeah.
I think we got it. Okay. So, for our main talk, we have Gene Hébert. We tried to pronounce it. Okay. And I met Gene at a dinner a few months ago, and he was doing really, really interesting research on like replacement. So like there's this interesting thing, you guys have all heard of Brian Johnson, like I think at a high level it's like you eat broccoli, you'll live forever. I think that's like probably not gonna work. We're gonna need more sophisticated scientific interventions. And like he was telling me about this really interesting idea of like, you know, you can replace the organs, you can replace your hand in the future, you can suppress the immune system, but like the brain is, you can't replace the brain, you're Frankenstein. And so we've been in contact, and he's written a really interesting book, which you can purchase on Amazon, and I will send out the link, and I've been reading it over the past week or so, and it's actually really good. I've been learning a lot about it, and it's back there, I should have brought it up here. And he's a professor at Einstein Medicine, and has a lab, has started a company that is doing brain research, and I'll let you take it from here.
Great. Thanks, Mike.
dealing with here. Let's say you could have a young body until you're 150. Who here would want to live to 150 with a young body? Oh, good. Good start. Okay. What about indefinitely?
Hell yeah.
So, I guess maybe the first part of what I'm going to say might sound like a bit of a downer, but there is hope, I think, with some of the approaches that a fraction of the field of regenerative medicine is working. So, yeah, progressive brain tissue replacement. This can be interactive too, so please feel free to interrupt, ask questions. But yeah, the first point I want to make is that aging is a difficult problem, and most people in the longevity field completely ignore it. And the reason they ignore it is because either they don't know about this data that exists from decades ago, biochemists characterizing the complex forms of damage that occurs in our bodies to our macromolecules, so those are proteins, carbohydrates, DNA, lipids. And instead they just focus on other things that change with age. But everything changes with age because the environment of the cells in the body is falling apart. So this slide is just meant to illustrate some of the examples that these biochemists discovered long ago. And they gave up on aging because they said, there's no way of dealing with this. That hasn't stopped a lot of people today of trying to combat aging with different approaches. But really, the only approach that has a chance of working is replacement at a tissue or greater level. And again, it's because these are just examples of different classes of stochastic damage that occurs just to proteins over time in your body. It's a physical process. We have no genes in our genome that recognize any of these forms of damage, or much less can process them and get rid of them. So all these genetic approaches, these epigenetic approaches, which is maybe some of you have heard of partial reprogramming, there's no way that that can address without reinventing a genome, which we're nowhere near being able to do. There's no way that can address these forms of damage. And that occurs in the brain, but in all parts of the body. So this is sort of the downer side of it. But there is a way, right? And that is replace it, right? So what if, we could take your old body and just surgically replace it with a new body, a young body. Like if you were 80 and replace it with a 20-year-old body, you would then be 20 years old, biologically, right? Replacement will work, right? So it's just a question of making it work. The one biggest challenge, I think, people are actually working on whole body replacements. I think that will happen before too long. But it won't do much good if your brain is still aging in that whole body, right? You'll have dementia by the time the healthiest, most intellectually sharp 100-year-olds are nothing like they would have when they were younger. So your brain ages no matter what. So you need to do something about the brain. And the challenging part, of course, is the brain is who we are. It defines our personality. It houses our unique memories, our personalities. So we don't want to lose that, right? That would defeat purpose. So is there a way that we can make the brain young again? And I'm going to argue that there is, and I'm going to tell you why. And of course, it's with progressive replacement, not with, I can probably manage with other slides.
We could try the other dongle.
Yeah, if you want.
Let's try.
Sure.
Because this doesn't seem to be working.
So I don't know, some of you may be familiar with the Shipathesius mythology, where, you know, you take an old boat, you replace it plank by plank, and then we don't know. I guess the question there is it the same boat? But for the brain, you know, as far as we can tell, the evidence is that it would be the same. So I'll give you a couple of examples that I've come across in the clinic. I'm not a clinician, but talking to clinicians, where they've seen individuals in their 50s, 60s, 70s with benign brain tumors that have eaten away at either their language center, they occur randomly in the brain, or the frontal lobe, which is a big part of personality. And these benign tumors start as a single cell and grow out and eat away at the tissue. Eventually, they have a headache or a seizure or something. They go to the hospital. Doctor says, you know, good news, bad news. Bad news first. You have brain tumor. Good news, it's operable. They take it out. Now they have a big hole where the tumor was. But they didn't change. They were the same person they've always been. Right? Their personality, if it's in the frontal lobe, didn't change. They never lost the ability to speak. No one noticed a difference. So the reason is that the neocortex, this is the part of the brain we're tackling first, the biggest part, the most important part, in terms of defining who we are as individuals, is incredibly plastic in how it encodes information. So if you give it time to relocate a function that is being used, it has to be used or else it'll be lost. If you give it time to relocate a function that's being used, it'll do that. Right? If you get a stroke in the frontal lobe, it's a different story. There's no time for that information to be re-encoded elsewhere in the neocortex. Personality changes. You get a stroke in the language center, you lose the ability to speak. Right? It doesn't come back. So that's one aspect. So I kind of jumped ahead here without the slides, but you know, plasticity. Here's just the example. Most clinicians are a pain in the ass because they don't publish these amazing findings. So, you know, at least some of them do, and make a study of it and compare all these different patients, same age, same amount of damage from stroke versus slow-growing benign myeloma. Thankfully, you know, we have, but if you talk to clinicians, you know, many of them come across these examples without publishing it. The other thing that's encouraging about progressive brain tissue replacement is there's dozens of labs, including ours, who have put in brain precursor cells in the adult brain and shown that they differentiate into neurons that connect surprisingly well to the rest of the brain. This was a surprise. Some of you in the neuro field may have been familiar with the dogma that there's no new axon growth in the adult, and that's absolutely true from existing mature neurons. But if you put in these precursor cells that give rise to immature neurons, it's highly reproducible. Everybody sees the same thing. They project to distant parts of the brain, not only that, but to appropriate targets. We're not sure how they do that yet. They may follow signals that are still there from development, or they may just be following, you know, remnants of axon tracks that were there before, but in any case, they do it. So new neurons can connect efficiently with the brain. And maybe I'll give you an example from my lab of that as well. This is not the example I was just alluding to, but another example where we put in, you know, precursor cells for the neocortex, and so here's the graph. It's human in the mouse cortex. It still works. We just have to immunocompromise the mice, and you know, you can see all these axons coming off the graph going down into this fiber tract. That's what they're supposed to do, and they do it. Even though we haven't come up with a graph yet that's functional, it's still sort of a jumble of cells. We need to fix this. That's what we're really focused on. But the neurons are doing what they're supposed to do, and they'll project and connect to other parts of the brain. And again, we're not the only ones or even the first ones to have shown this. You know, all these high-profile papers with pretty spectacular titles saying that, oh, wow, we've repaired the brain. No one's come close to repairing the brain yet, but you can sort of repair at a very base level circuits in the brain. So those two things, the fact that you can slowly remove old tissue without losing its information and add new tissue that can encode information means that we can progressively replace brain tissue in theory without loss of information, without loss of personality and our memories, as long as we're recalling those memories during the silencing process before removing tissue. Okay, so why haven't we done this yet? And why doesn't it work yet? So there's a couple of reasons. Again, a lot of labs are doing this, but progress is super incremental. Again, this is a whole other thing we should talk about, is the way that science is done now. Small incremental progress is rewarded. Trying to reach further than that, you get slapped down, no funding. So things have moved very slowly. And so the biggest things that are missing so far is that this is like one of the better looking graphs in the literature. And there's no architecture here compared to normal, nice layered tissue here. There's different types of neurons. That organization is very important for the intra-graph, or would be important if they had a graph that was layered, for intra-graph information processing. There's a whole flow of information that occurs in the cortex between the layers, and that's important for information processing. That is not recapitulated here. There's also important cell types missing. There are many types of neurons in the cortex, and a lot of those are missing from current graphs. And they're very, again, important for feedback information, feed-forward information. So they're needed for learning new things, and they're missing here. So we're not there yet. The approach that we're taking is one of trying not to reinvent the wheel. Brain tissue, one of the most complicated things we know of. And we wouldn't be able to put in mature tissue into a person. For one, it's too complex to make. And two, it wouldn't integrate. Again, because mature neurons don't project their axons and connect to the rest of the brain. So what we're doing is just mimicking the way our brains were made in the first place. Right? No scientist or engineer made any of our brains. They all developed from a relatively simple tissue early in fetal development. Right? So this is a much more manageable task, is to reverse engineer that simple tissue. Right? We found a stage, it's kind of a sweet spot where there's enough internal structural cues and signaling cues for that tissue to develop on its own, and yet it's simple enough that we can engineer both the cellular components and those extracellular components. So that's what we're trying to do here. This is just an example. So in doing this too, we use like high resolution analysis of real human fetal neocortical tissue. Not everyone here is in the bio field, but we look at proteomics, we do single cell sequencing just to really find out what's there and then mimic that. And so, it's been fun from a scientific point of view because we've discovered, molecules that no one previously knew were there. And they seem very important because they can be very localized to structures that we know are very important. And specialized cell types that we know are very important, but we don't know, how they get there. So, it's fun from a scientific point of view, but of course, we want to be able to engineer this tissue. So these, I won't go through this, but basically these are the precursor cell types that we need for the tissue we're engineering, and also the extracellular components. So we're at a stage now where I think we've identified everything that we need to make this tissue. We can generate all these cell types from human iPS cells. So I don't know if you guys are familiar with iPS cells, but you can take cells from your blood or your skin or other parts of your body and bring them back to like the earliest embryonic stage, and from that generate any cell type in the body that you want if you develop the protocol to be able to do that. But they have the potential to do that. And so we're at that stage where we can generate all but one of them. So we're almost there. Then the trick will be to assemble them and test their function. So we have a platform for doing that in mice, as I showed you earlier with the human tissue. So we basically just make these very reproducible lesions and then put our cells with their extracellular environment or their biogel or niche, however you want to call it, into that space. And then we look for how well it functions, how well it develops in a structural way. And sort of a couple of highlights, these are from papers we've published, but one illustrates the neuronal integration I mentioned before, where axons project to the right place. They also receive new inputs from the surrounding brain, and this is an example of that. So in this case, we put new, proto-tissue. That might be an exaggeration. We put new cells and biogel in the lesion site, in the visual cortex in this case, and wait over time, and find that these neurons, if we're recording from them with an electrode, we can detect their activity. They pretty much behave like visual cortical neurons do in a newborn, mouse in this case. So initially, there's just sort of spontaneous random activity, even though we're exposing the mouse to a visual stimulus, which is this little thing here. But eventually, those neurons start responding specifically to that visual stimulus. And then they get very good at it. And not only that, but they get tuned to very specific visual stimuli, like normal neurons do. So again, very encouraging, but we're not there yet. This response does not mean that that tissue is processing the information in a useful way and sharing it with the brain so that it contributes to a useful behavior or function to the mouse. But it's encouraging that we can get there. The other one is vascular integration, also very important. So we include, we have the luxury of genetically modifying the cells that we put in, so we'll make them fluoresce green for our vascular endothelial cells so that we can compare them to total blood vessels. And we find that they quickly form vessels after grafting. These vessels fuse with the host vasculature, and we even have videos that show that you have little red blood cells going through there, and you get circulation through there. So you can get vascularization of these grafts very quickly, which is important when you're considering that you want the structure to stay stable after you put it in. So those are just two examples. So I guess just to take a step back now and say, how would this work? So you could potentially use this to repair brain tissue, and that would be our first clinical indication would be something like stroke. And that's what I started a company for, to accelerate progress towards the clinic for a proof of concept that our tissue works. But ultimately, since all of us here want to live, at least to 150, that means we need to fix the brain. So the way that would work is with this progressive tissue replacement. So initially, the brain shrinks, right? So it does on its own create room for new tissue. So you could put in this new fetal-like precursor tissue that grows into a mature tissue as it connects with the rest of the brain and starts adopting function. And while you're doing that, you can start silencing other parts of the neocortex. Right? And you can potentially do that not with the example of a benign brain tumor I told you about, because they unfortunately sometimes give rise to malignant brain tumors. But you could do it with existing optogenetic approaches where you can use light to silence neurons from a pinpoint out, as a tumor does. While the person, you can first map what the functions are there, which is what neurosurgeons do when they do brain surgery, and figure out what is roughly encoded there, and tell that person to keep thinking about that during the process, and it will get re-encoded elsewhere, and they will not lose it. And then that will create more space for new tissue, and you repeat the cycle, and you could potentially have a 20-year-old brain again, but with all your memories from a lifelong living. So that would be the idea. Sure, please.
It's just kind of optional whether or not you want to keep the memory. So would you
Yeah. Yeah, no, good
erase
point.
a memory like that?
Yeah, sure. Yeah, and you could also probably try learning a new language when you have new tissue anywhere near the language, so acquire that like a 2-year-old acquires it as opposed to how we currently do, which is kind of painful. So how does this fit with decentralized science? Not sure yet. It's a big enough project that I wish it did. I wish we had a lot of people working on the different aspects of this approach. I only talked about the neocortex, but what we learn here in part can be applicable to other parts of the brain which nobody is even working on yet, but they are also essential to normal function, and we would also need to address them in aging. So we really do need a sort of this collaborative community of scientists working on this to get there in a reasonable amount of time. I'm very open to that. The first people I talked to were a DAO, Vita DAO. Some of you may have heard of them, but they're a longevity DAO. And I love the idea of getting everybody involved and more people involved for working on this. So they actually fund our company to some extent. But we need more of that, and certainly at a scientific level. So we need more of the people trying this in different places, different ways, sharing the information, what works best. We'll move forward a lot faster. The way we're doing it now, we think of what we need. Okay, there's like 10 different ways we can get there. It's like, well, we only have 2 people working on it. We can only try one of these. If we fail, we got to come back, try again, fix it. It's going to take a long time. But if we can be working as a community, different people working together, exchanging the information, we'll get there faster. I don't know how we get there to do that, but we'd love to. Yeah, that's pretty much it. That's just the company. So combined with body replacement, which, as I mentioned, other people are actually working on, and it's technologically not that difficult. If you guys want to hear more about it, happy to tell you more about it. But if we can do that and the brain, we actually can beat aging and decide how long we want to live. So I think that would be cool. Yeah.
Just have one question. What kind of inflammatory response do you get when you replace or sort of introduce this new brain tissue? And do you need to treat the animal models with some sort of immunosuppressant drug like tacrolimus or?
Yeah, no, absolutely. So there's sort of 2 parts to that. There's there's always inflammation whenever there's a type of lesion for aging itself. Lots of inflammation because there's lots of damage. But certainly in the way we do it, where we actually remove tissue, it's surgical removal. So there's a lot of inflammation. We use anti-inflammatories, methylprednisolone, and that helps. But we occasionally still get some scar tissue, so it's not perfect yet. There's more we can do there. Then there's not having the host reject that graft. And so right now that involves immune suppressants. So there's brain cell therapies in the clinic now for Parkinson's, for epilepsy, with very good early results. But they're also using allogeneic cells, so not the patient's cells, but somebody else's. And so they take immune suppressants as well.
With autologous cells, would you, do you think, even if they're coming from the patient, you still see tissue rejected? If they're coming, you know what I'm saying? The patient themselves, right?
There's not too many examples of that in humans yet, but so far the ends of suggest that it's not a problem, but it's still early stage. But there shouldn't be, right? They're genetically matched identically. So that's the idea. And there is a company on the West Coast who just got FDA approval to do that for Parkinson's with patient-derived, iPS-derived dopaminergic precursors to treat Parkinson's. They don't have results yet, but the ones that are using non-patient derived are getting good results for their Parkinson's patients. That's a group here in New York, founded, the company is Blue Rock, founded by Lawrence Studer. Big fan is.
How do you approach the replacement in stroke where there's acute disruption of structure and function, whereas the approach you showed is more progressive integration of the graft into
Yeah, there's not a quick and easy answer to that question. It's a good question. It depends how long after the stroke you plan on putting the tissue. If you wait long enough, you now have a depression and space. If you do it in an acute inflammatory stage, it would be a very different environment. So maybe somewhere in between would be ideal, but you still have like a scar tissue there. So we've consulted with neurosurgeons, and that's actually what they worry about most, is displacing the scar tissue to put in the new tissue, because that might cause more bleeding. They're very cautious. Despite the fact that neurosurgeons are like the craziest MDs out there, because unlike other doctors who, you ask them about a potential treatment, they say, oh, has somebody else done it? No, then I don't want to do it. A neurosurgeon, if you ask, has somebody else done it? Oh, that's boring. I don't want to do it. They want to be the first. So that's good for us. But so that's actually, a question that we have to be very careful about, conscious of when we're going to put in new tissue. It's how are we going to do it without causing too much bleeding. I mean, there are things you can do to mitigate that, but still, you want to really reduce those risks.
So you mentioned that you were focusing on the neocortex, and then you had also mentioned this, I think it was like a 5-week sweet spot of development, where it was at the right area to engineer. So is that development process and timeline, is that uniform across the neocortex, or does there end up being regions where it differs?
There are gradients of development across the neocortex, so we're kind of, talking about a generic middle for that age. Human neocortical development is pretty protracted, so it's not as dramatic as you'll find in analysis. Those gradients are easily seen looking at by eye thickness or things like that. So it's not, so they are there. We should be conscious of them also because there's some predispositions to function that seem to exist early on in cortical development. We don't know if it's that early, but
so it may be possible that you would need different starting points for different regions, but you're hoping it can be universal.
Possibly, yeah. I mean, certainly, around the edges of the cortex, the canonical layered structure breaks down. The archicortex, cingulate cortex, those are clearly different, and we would use, a different precursor structure for that. Good question.
In the future, do you foresee this being something that could be covered realistically by insurances, like for treatment of Parkinson's, Alzheimer's, dementia, or is this going to be more a treatment for the rich and famous just to remain young?
Yeah, I mean, I actually just proposed this program to ARPA-H, and I'm waiting to hear back. They invited me for an interview, and so, they're really concerned about that. So I prepared a list of arguments for why it could be. One is that cell-based therapies, have already been approved by the FDA and covered for Medicare. Those are all blood diseases. So there's precedence for getting insurance coverage for those things. But there's also, really the cost-benefit analysis. So treatment of stroke patients and traumatic brain injury, which would be another target of this, are like $100 billion in the U.S. alone, or approaching $100 billion. And we would actually save society a lot of money by getting those people back to work and not having them rely on this lifetime of support care, which ends up being much more expensive than the surgery. So there's economic reasons why it would make sense. Whether we can convince people of that, I don't know. But yeah, I mean, it should be for everyone.
Isn't that the point of decentralization?
Yeah. Exactly. Yeah. Yeah. Sorry. I think he's had. Who's first?
Okay. Either way, sure. Thanks. Yeah. So I was wondering with. A lot of people, older people, have protein aggregation in their brain, and people talk, and the theory, the dominant theory is that it spreads through this like prion-like mechanism through the brain, and maybe once it gets to a certain point, it may be caused by aging, but once it gets to a certain point, it may become sort of self-sustaining. So I wonder, do you think that these graphs are going to be especially in cases like Parkinson's, where there's like a protein aggregation nature to it, do you think these graphs are just going to be destroyed by that process after a while, or are they going to resist it?
I don't think so, based on the evidence so far, which is, they were doing these transplants using human midbrain tissue to treat Parkinson's in the late 80s in Sweden, and some of these patients were essentially cured, some were made worse, so they had to improve before they went back to it. But in some of the patients, 2 decades later, they did post-mortem analyses and saw that the neurons were still there that they transplanted. So, and those are naked cells, right? They weren't part of the tissue. So ours are going to be much more insulated from the environment. I'm not saying this is going to be true for all aggregates. Prions, maybe a little more problematic than AD aggregates or other aggregates, but I think the evidence is in our favor so far. Yeah. We are looking at that too.
I guess that's something, if you had like 100 extra people and some DeSciNYC funding, you could test in mouse models and that sort of thing.
Yeah. Yeah. Do you still?
Oh, I had a question. In terms of Parkinson's also, I know that stems from the midbrain. Could the graphs be applied in the midbrain, or does it have to be in the cerebral cortex?
Well, so the graphs now that these companies are in clinical trials for getting good results, similar to the early studies in Sweden, they're putting the cells at the target location of where the neurons that are lost would have projected to, rather than where the neurons were lost. And that's because back then, they didn't realize that young neurons can project in the adult brain. It's been shown in humans for Parkinson's patients postmortally as well, that you get these projections. But again, because they got these positive effects, no one sort of wanted to take the risk of trying to put them where they belong instead of where they have been putting them, because there was a positive effect. So they've kept putting them in these places. So the current clinical trials, they put them in the striatum, not the substantia nigra. And there's one guy that had the courage to at least put them in both places. So he put them in the striatum and the substantia nigra. And in postmortem patients saw that they had connections. They couldn't tell which direction they were going in, but those are bidirectional connections anyway. So even in the human brain, in the adult, in a diseased environment, you get proper connections. So that's very encouraging as well.
She had a question. Mine's kind of dumb, so if you want to.
Oh, yeah, sure. So I'm just curious. So since the aggregation of proteins like tau, alpha-synuclein, and amyloid can start accumulating in your 20s, 30s, and 40s, I'm just curious if you diagnostics of like amyloid PET, tau PET, if you can diagnose some of these neurodegenerative conditions earlier, and if you use the tissue degeneration, can that help prevent the onset of these neurodegenerative processes?
Maybe if, the really early cases are usually familial, so genetic. Right? So in those cases, if you can identify the genetic lesion, you could, even in autologous patient-derived cells, correct that before making tissue and replacing it. So potentially, yes. Yeah, like ApoE4, for example. I would correct that if I have it.
It's, well, quickly, thank you for your talk. This is my first time at one of these. This has been highly fascinating. Fortunately and unfortunately, some of the awesome people here asked like questions I already had, so I'm left with the sadly philosophical one that kind of want to ask. I'm following up what you said about insurance, where while I know it's the early stages, and as someone who's done research before, even in computing, it's very hard to get off the ground and implement stuff. Do you or any of your researchers sometimes step back and, including all the wonderful things to get used for, do you sometimes go, oh God, this is going to change a lot of things that I hope like society and legal stuff will hopefully catch up to if, for example, humans become deceased ships such as that. One quick example is we base a lot of our social political stuff on our own term limits. So there's a lot of positions in our society where you just run those until you're dead. So if you're able to even go on to advanced age or live forever, that kind of becomes a problem.
Yeah, a little bit, you know, you can't help but think about these different things, but always weigh that against the fact that aging will kill everybody on this planet if we don't do it. That seems, if you value life and, you know, an individual's life, that seems to outweigh pretty much everything else. Those other things, we can adapt, you know, we can change the way we do things. But, you know, to me, that balance is always the fact that we're all going to die is kind of outweighs.
I will say personally, while I could be here all day and like disagree very fascinating with different things, I do agree that I love Jesus research to like someone's dying and please help them. That's like the ultimate thing right there. So yeah.
Great tips.
Within your field, where do you think like your colleagues are wasting the most time?
Getting IRB approval.
Besides admin, obviously.
Yeah, there is a lot of bureaucracy.
Line of inquiry wise.
Line of inquiry wise. So which field do you mean exactly?
However you want to self-define, you know.
Yeah.
Like when you go to a conference and you lock eyes with the guy across from you and you like, you know, you scorn him. Like, what is he doing wrong?
Well, yeah, I mean, I'm already taking my punches at the longevity field. I think that, you know, when I go to a conference on longevity, that's pretty much most people there are wasting their time.
You don't want to drop some shade here.
Yeah, I don't know. Let's all work together.
Well, thank you. Thank you for coming, and thank you all. You can stick around and chat for a little. And thank you again to our speaker. That's really and do you have a way to get in contact with you or something?
Yeah.
Okay.
Pretty easy to find.
Okay. Google his website.
Google my name and add Einstein or my institute.
Sweet.
Yeah.
All right. Well, thank you guys, and make sure to come to the one on the February something on ultrasound. Thank you guys, and stick around.
Woo!
Hey.
How you doing?
Good, how are you?
Good. All right.
Good job, Michelle. Participating.